Welcome to day two. I'm very happy to very briefly introduce Laszlo Barabasi. He doesn't really need much introduction. He's at Northeastern after having been at Notre Dame and various other places, is clearly a giant of network science and network theory. And so we're going to hear about control in physical networks today. So I'm looking forward to
Perfect. Thank
what
you.
Laszlo's up to.
Thanks for the opportunity of being here. And let me just prepare for the job. and indeed, the challenge was for me is like, what would be the most appropriate for our discussion today? So I decided to talk about this project, which is really kind of, as we heard, everybody knows my lab for the work. Okay, so I'm sorry, this keeps kind of kicking me into the internet, so I'm going to have to turn this wall off and turn off the internet. So anyway, so my lab is mostly known actually for focusing on the structure of large networks. And really, I think the network science community has achieved quite a bit of advances in that space in the last few years. But one of the questions that I've been always asking is that what kind of knowledge have we actually gained in this particular space? And in particular, what did network science offer us? Well, it mostly kind of looking at very large networks and understanding them. And understanding them meant actually that really we try to quantify and eventually predict the behavior of the system. Now, the challenge with actually that we've been focusing in kind of about the last 10 years in the lab is that how do we go one step further? Because eventually the reason we want to understand these systems like the internet or like the subcellular networks in our cells is to be able to control their behavior. And so the control has been a central problem in my lab in the last few years. And the question is, what do we really mean by control? And of course, we're kind of borrowing the language and the tool set from control theory and taking it in the context of complex systems, what we're here to talk about. And control theory is a well-developed discipline within applied mathematics. Most control theorists actually sit in the electrical engineering department these days. And I'm going to pick one particular angle of control theory, which is the issue of controllability. So what is controllability? Well, from a physicist's perspective, controllability is an ability to take a system who is somewhere in the phase space and move it somewhere else in a finite amount of time. That is our ability to change the system's state in the phase space from point A to point B. And now, of course, this sounds a little bit abstract. I'm going to put this down. And let me give you an example of how this would work. So I'm showing you here a car, and the reason I'm showing you is because a typical car is a pretty complicated system because it has about typically starting on 6,000 different components. Yet, when we drive a car, we are not thinking of all the 6,000 components that the car has. Rather, we are aware of 2 or 3 components, the gas pedal, the brake, and so on. And by manipulating these 2 or 3 components, we are able to move the car from anywhere to anywhere where a car can go, right? So we can move it within the phase space available for the car. And the reason we can do so is because the car has been engineered to be controllable from 2 or 3 input signals, which is the steering wheel, the gas pedal, brake, and so on. Now, so much so that I urge you never buy a car that would lack that particular property. But on the other hand, what if I revert the question? What if I'm going to ask you, here is a wiring diagram of a car. Tell me what are the components I need to control such that from there I can control the rest of the network. And you may say, why would I ever bother answering that question? Because the answer is known to engineers. But yet I would argue that this is the type of question that most scientists face on their day-to-day work. They're faced with a very complicated network that somehow nature has produced, generated, evolved. And the challenge for them is to understand what are the components through which they can actually control certain behavior aspects of a system. For example, you know, a few years ago, the Nobel Prize in Medicine has been given for the discovery of 4 genes whose state, if you changed, you actually turn a normal cell into a stem cell or stem cell into a normal cell. What was that discovery? Well, that discovery was really finding the 4 control points of that particular transition in the system. So what I would like to do today is to ask the question, could we actually find the control points for an arbitrary behavior of a system? And in order to do so, we have to model the system. And we're talking about the network. So any model will have to start actually by looking at the network. And here's my little network, right? And all right. So that's my little network over there. And effectively, it's not enough for us to have a network. We will also have to have some dynamics on the system, because behavior implies there is some dynamics happening on the system. So in this particular case, I'm going to define a variable x on the system, this one, on each of the nodes. And there is some dynamical equation that tells me how these variables will change. And of course, we assume that these variables will change mainly because the node interacts with other nodes and gets signals from the other nodes. And that's kind of would be the self-dynamics of the system. But we also allow for the possibility of control. And the way control is formalized is that I have the possibility to inject signal from outside inside the system that is a time-dependent signal on some selected nodes. So fundamentally, I have an adjacency matrix that describes the network. I have a dynamics that is the self-dynamics of the system that here I choose linear, to be linear. And I'm sure many of you will ask what happens for nonlinear. We'll have an answer. And then the B vector just simply tells me where the signal arrives and what kind of signal will that be. Now, the question I would like to ask is that, am I able to control the system? Well, if I put a wavy line on every single node, I can control the system because I can put it in the position I want to be in the phase space. But the challenge is, can I control by really giving signals only to these 2 nodes and not the rest of them? And the answer is given by the Karman condition, or the wrong direction. And this was actually the work of Rudolf Karman from the 1960s, in which he actually showed that if you have the 2 matrices, B and A, you know, where the signal comes and what's the network, you can build a controllability matrix C. And that controllability matrix here, the rank of that matrix that you build from that will tell you whether the system is controllable. If n is your number of nodes, if the rank is equal with the number of nodes, you have a controllable system. If it's less than that, you're not able to control the system. So that's kind of the theory. And let me just illustrate how that works. So let's start with a little network. And I'm asking now, could I control this chain of nodes from the top? And the Karman condition will say yes. And the reason why it will say yes is because we actually have a formula for control theory of what kind of signal you would send in the top, so that taking advantage of the delays on the nodes, I can set the time dependence of the remaining nodes in an arbitrary state. So this problem is very well solved. How about this one? Doesn't look any different. 3 nodes, 1 signal. But control theory tells me that this is not controllable. And the reason why it's not controllable is because x2 and x3 will be always coupled. I'm not able to control them independently. So in order to control the system, I will need 1 more signal, which is break the symmetry, and then I have control over the system. The reason I like this example is to illustrate that control is not simply about the ability to the signal to arrive to every node, but the ability of unique signal to arrive to each of the nodes. And that's what really the controllability condition is checking through the path structure. Now, it looks like we have solved the problem because we have the Karman condition. But there's many problems with the Karman conditions. First of all, you need to know everything about your network weights and so on. And that's actually the least of the problem, because control theorists have developed in the 1970s the concept of what we call structural control, which is a theorem effectively saying that if I have a weighted network, I can replace the weights with random numbers, and the outcome of the Karman condition will be accurate apart from measure 0, meaning that if I choose my weights in a weird way, I could get the wrong answer. But that probability is close to 0. So the weights don't really matter, at least for asking whether the system is controllable or not. The real problem is this. The Karman condition doesn't tell me what are the control nodes. It tells me that if I made a guess, whether I could or not control the system from there. So it means that I have to try every single combination of nodes as input nodes until I arrive to one that actually controls the system. So that makes it practically an NP-complete problem because I have to try every single pair, triple, and so on. And the third problem, which is equally severe, is that we like in complex systems networks with 1 million nodes. But you can't even check mathematically the Karman condition for more than 1,000 nodes because you will have to invert an n square m matrix. N is the number of nodes, m is the number of links. And no computer will do it for over 1,000 nodes. Or you'd have a supercomputer to go a little bit further, but there are severe limitations. So we have a theory to know whether the system is controllable or not. In practice, you cannot do it for large networks. So really our question, what we ask is that if I give an arbitrarily large network, how many nodes do you need to control the system? How do you find them? And eventually, what determines their number and location and so on? So to answer that, I'm going to take now a detour and go in a completely different direction, which apparently has nothing to do with control, but obviously it does, which is a wonderful mathematical game called matching. Matching is like a little challenge. I'm going to give you a small network, and I'm going to ask you to color links such that don't put ever 2 links who end up on the same node. And so like, for example, this is a matching, right? None of the red links or red bonds that I put in the system are actually touching each other, ending up on the same node. And of course, if I want to make it a little bit more difficult, I'm going to ask for the maximum matching, the maximum number of red links, so that never 2 links end on the same node. And this is actually a well-known problem, both in mathematics. Graph theorists have actually focused a lot on that. Laszlo Lovász, who he and I are running an ERC project now in Europe, has actually even written a book about this problem. And it's a major problem in economics, because this is the one that, for example, this is the algorithm or the set of problems that medical schools use to match candidates with the school where they're going to do their residency. Effectively, what you have a list of candidates and their rank list, and you have a list of hospitals and their rank candidate list, and you want to find the maximum matching such that everybody gets matched as close as possible to their desired position, but you're also optimizing the system's matching properties. So that's what the motivation is. This has gone so far that in Boston City now, even the kindergarten placement is done by a matching algorithm, which has to do with the lot that there are too many MIT graduates in the neighborhood, right? So I'm going to generalize this one to directed networks, because in control, direction does matter. And it's going to be the same question. I'm going to ask you to find, to color the bonds, but with the restriction that on one node could be only one going in and only one going out. But you cannot have 2 going in or 2 red bonds going out. And then you will check that the maximum matching in the circle case, everything is matched because none of us is violating that principle. On the little star network, only one bond can actually be colored red. And one more definition, we're going to call matched a node to which a red bond is pointing to, and unmatched those that it don't point to. Detour is over. Why am I telling you this? Because we were able to prove a very simple but very powerful theorem, which is to say that this problem and the control problem are fully equivalent with each other, in the sense that the driver nodes for the control problem are the unmatched nodes. That is, anybody who is unmatched, who is not color green, must be controlled. And you can intuitively feel why that has to be the case. If a node is matched, then this node is controlling that. You don't need to control that. But those who are unmatched must actually be controlled from outside in order to control the system. Let me illustrate how this works. You give me a small network. I'm going to find the maximum matching. And I'm going to say everything who's not matched must be controlled. That's where your signal is going, right? And you will notice that there is redundancy in the solution, and in the way that I can actually choose the unmatched nodes in many different ways. But what you may also notice in this figure, that the number of control nodes don't change when I do that, right? So the redundancy doesn't alter the number of control nodes. Now, this theorem is very powerful because it takes one problem set, which is the control theory problem, and maps into a very well-studied problem in graph theory, the matching theory. But it may sound like we haven't really resolved much, because to find the maximum matching looks like an almost impossible problem to solve. So are we back to NP completeness? No, we are not. And the reason we are not, because what we know about the matching, that it is a polynomial problem. And there are very, very effective algorithms, square root of n times L, to really find the maximum matching in the system. So therefore, it tells us the control problem is not NP complete either, but it's polynomial. And when you look at it, you will find that with this scaling on your laptop, you can run a million node network, and you will find the maximum matching in a few seconds. So how does this work? Let me illustrate this. This is actually a network of within a company in Hungary of like who is listening to whom, who's talking with whom, this is actually a project we did many years ago of mapping this out for many companies. And it's a directed network. And if I apply that, I want to find a bunch of nodes, those who are vibrating who are not control nodes in this system, but there are a few who are actually control nodes, and you can very easily, the red nodes are the control nodes, and you can very quickly, easily identify them. So now we have a hammer. We have a tool to identify the control nodes. We haven't still answered the question, what determines the number of control nodes in the system? And for that, we started to look at a bunch of real networks, regulatory networks for World Wide Web and everything, not because we want to control all of them, but because these are examples of directed networks whom we could actually explore and study from the control perspective. And what I'm showing you is that what fraction of the nodes I need to control this system. That's on the axis, and these are different networks. And you see they go from almost 1, which means most nodes need to be controlled, to almost 0, which means that 1 or 2 nodes can control the rest of the network. So the question is, what network properties determine how many control nodes you have? And there we had a very lucky break. And the lucky break started like that. So, one of the things that we have to always do in network science is to say, the signals we get, is this really connected to network topology or could come from any random network that we are actually exploring? So what we did is that we ended up taking the real networks and randomize it, meaning keeping the number of nodes and links fixed, but otherwise changed who is connected to whom, effectively taking that network and turning into an Erdős-Rényi random network. And then we asked, for the same network, how many control nodes did we need originally, and how many do we need now in the randomized version? And of course, the two numbers were totally disconnected from each other, not surprisingly. You totally destroy your network, all the signal is gone, you're dealing with a random network. But then, of course, we have to be a little bit smarter than that, because we know in network science that complete randomization makes no sense in most cases. What you need to do is to do degree-preserving randomization. That is, if you are a hub, you want to maintain your status of the hub in the new system as well, in the randomized version. And if you are a small node, you're still going to have, say, only 2 links, originally and in the new network. That's what we call degree-preserving randomization. And the big surprise, numerical observation, was that the real and the randomized network had the same number of control nodes, which was really odd because the network was gone except the degree distribution was maintained, which is telling us that the number of control nodes, therefore, is determined mainly by the degree distribution of the system. Now, that's a lucky break. And the reason why it's a lucky break is because effectively it's telling us that if I know the degree distribution, I should be able to determine the number of control nodes. And actually, we could use the cavity method to do randomizations in the system and analytically calculate the number of nodes, number of control nodes you need. And for example, we analytically derived that for a random network, the number of control nodes depend on the average degree by decreasing exponentially. The denser the network is, the less nodes you need to control, which kind of makes sense because if the network is very dense, there are lots of alternative paths, so it's easy to get unique signals to every single node. And even for a scale-free network, we could analytically derive it the same behavior and so on. So the cavity method allowed us to kind of go ahead and analytically describe the system and describe many of its interesting properties. Now, what was very interesting as a conclusion, to make a long story short, is that what we found is that the mean degree and the degree heterogeneity, how different the degrees are the two main factors that determine the number of control nodes. And in particular, we found that sparse and heterogeneous networks are harder to control than dense and homogeneous networks. And the reason why this is odd for a network scientist is because most real networks are sparse and heterogeneous. So therefore, what we are finding that real networks are very difficult to control just by the nature of their construction. Now, in the remaining minutes, I'm going to switch gears, and I'm going to say, all right, this is all a beautiful theory. How do we know that it works, right? And can we actually get evidence for that? And one way to know it is actually, for that, I need one more piece of theory, which I would call target control. Target control addresses a major issue in control, which is to say, when I'm trying to control a system, I don't have to control all the nodes. It's enough for me to select a certain set of nodes and control those, and I don't care about the state of the rest of them. So the target control effectively says, I have a set of target nodes, and I want to control them. And what are the control signals I need to build, and where do I need to control to achieve that? Why is this going to be important is because the reason is, and there's a formula that you can actually adapt the rank condition to solve that problem as well. We did that. And what I would do next is to go ahead and apply this target control principle, and I'll show you how, to a real system. And I heard there was lots of discussion yesterday about the brain, so why not to actually apply the brain to the brain? Let's control brain. Could we do that in an experimentally verifiable fashion? And of course, we are modest, so we're going to start with a simple brain, the brain of C. elegans. And C. elegans, as many of you know, is a very highly studied organism, and it has a brain with about 300 neurons. And with that, it can do things like that. If you touch it, it doesn't like to be touched. So it will sense the touch, and it will turn away and run away from you. And that's what you see in this, that, now it's being touched and turned back and goes in the opposite direction. The question is, how does the C. elegans sense this, and how does it change its behavior? What are the control processes that allow us to make this change in the behavior? And we know that because the C. elegans brain has been fully mapped out, and we know that there are sensory neurons. These are in this layer. Then there is a bunch of control system in between. And then there are eventually which are then controlling the muscles. And the muscles are the ones that execute this kind of new motion. And how do we know that these are the neurons involved? That you see there are specific neurons involved in this process. And we know that because 30 years of experimentation has led to one by one knocking out individual classes of neurons and figuring out whether that absence of that neuron or that class of neuron will alter that behavior or not. And what you see in this table is the summary of 30 years of experimentation. So if you knock out, for example, the AVB or AVA neurons from the brain of the C. elegans, you will get uncoordinated motion. It's going to feel like it's drunk, right? But there are others that actually, like if you knock out DA and DB, you will have loss of backward or forward locomotion. It will stop moving effectively and so on. So by painstakingly kind of knocking out these neuron classes one by one, the neuroscientists came up with this diagram that this is the pathway that is responsible for the control of the mobility in C. elegans. Now, this is a beautiful body of work. I have one problem with this. When you look in the context of network of these neurons, they are not like that nicely isolated. That's how the network looks like. Right? And these are the neurons that I showed you earlier, kind of like being part of that. And you see that these neurons are connecting to dozens of other neurons at the same time. So they don't exist like a beautiful pathway. So the question I would like to ask, could I start from this wiring diagram and figure out what are the neurons involved in this process? And we do so by using this principle that I showed you earlier. You remember we said this configuration is uncontrollable because these two are not independent, and this makes it controllable. Let's generalize that. If I have a layer of neurons that are trying to control another layer of neurons, if you have fewer neurons at the top, it's uncontrollable. If you have more neurons at the top, it's controllable. It's like a really bad kindergarten where you need to have more teachers than how many students you have in the class, right? And that's the only way you can achieve control. So let's apply this one to the C. elegans. And let me just show you where my problem is with that simple pathway. When you look at the connection between the sensory neurons and the muscle, this is the reality. The signal from the sensory neurons to the muscle goes through this many neurons, right? These are all the directed paths that are going through. So how could you really separate these very simple paths that we showed earlier? Well, we apply control now to these two layers, the muscle layers and actually the layer above it. And that particular principle, and when you look at it, you realize that you have about 9 neurons trying to control 15, so the system is not controllable, which is okay. I don't need to have independent control over my muscles to actually move my hands. Many of my muscles are deeply coupled to each other from the control perspective. So the question is not whether the system is controllable or not. It is not fully controllable, at least not independently for each node. But the question we ask, what if I knock out one particular neuron here? Will that affect how many muscles will I be able to control? And the answer is that in that case, it does, because the number of controllable muscles went down from 15 to 14. So good. So there's an impact of removing a neuron. But what is interesting is that if you do this exercise for every single neuron in the C. elegans, of the 279 neurons in the C. elegans, only 12 neurons have the property that if you remove that, it will actually affect the controllability of the muscles. The rest you can remove, it will not change the number of controllable muscles. And that's where the beauty comes in. When we look at who are these 12 neurons, we exactly and fully recover 30 years of experimentation in this field. And when I saw that, I said, oh, this is just cannot be a miracle, right? It has to be real. We're capturing something essential to how this kind of C. elegans brain works. And of course, if all you do is recover what is known, you know, that's exciting, but you want to do a little more. And it turns out that there's one more that the theory predicts that was not part of the experiments before. That is the PDB neuron. And what is PDB? This is kind of the setup of, if you look at a part of the brain, PDB is sitting somewhere here in the layer of 5. And if you remove it, you will actually lose control over some of these muscles. And this was a brand new prediction. So we ended up kind of teaming up with a group from Bill Schaefer's group in Cambridge, UK. And they have done what had to be done, kind of ablate that particular neuron. And they showed that the effect was exactly what we predicted and where we predicted, because it was affecting the motion of these neurons. And the way they do that effectively is they put under the microscope the C. elegans, they ablate this, and they look at the one that is not ablated, and they find differences in the motion, and those differences are actually rooted in the muscle. So, and I'll show you some examples. So here's when we end up. We have this full list. This was a brand new prediction, and it's fully verified by the experiments. But we could do even more. And here's a dirty secret that I didn't tell you until now. This is a single neuron, but these are all classes of neurons. There are sometimes 10 neurons within the class. The reason why they are classes is because they're morphologically, traditionally indistinguishable. They look alike. So traditionally, people knock out a class, the AVA class, all the class from there, and then ask whether it affects the motion or not. But in the last few years, the technologies got precise enough that they can knock out individual neurons. So we took one of the classes, the DD class, which consists of 6 different neurons, and we said, which of the 6 are responsible? And we predicted 3 to be essential, those 3, and we predicted the other 3 to not to matter at all. And of course, they did this experiment as well. They knocked out one of the essential we predicted and one of them that we predicted not to be essential. And the result was right on the money, that it worked how we planned. And to give you an intuition, and that's where I'm closing my talk, this is how a normal, healthy C. elegans moves. And just to train your eye about what it is. And now I'm going to show you the one that has actually the DD5 removed. And the first may not look any different. Oops, that's not what I intended. So let's try again. OK, all right, that's the healthy one. And let me do it this way. And that's the sick one. And it doesn't look any different. It's not about the speed. But if you see, the back is not curving. And that's exactly what we predicted, that the DD5 will affect the controllability of the back muscles in the system. And of course, the experimental evidence is not by looking at the eye, but actually by looking at the eigenvectors of the mobility and figuring out that there's a difference there, statistically significant difference. So this is kind of how you actually can see. In some of the other predictions, you can't even see by eye the difference. So let me conclude here. And the reason I'm very excited about this project, because we all come from the complex system field, and we're all excited about scaling locks and the large-scale patterns of the system. But when it comes to biological systems, what distinguishes them from many other systems we study is that they have function. And we have had, or at least my lab has had, difficulty in the past to get to this function. And I think one of the reasons we had difficulty for that is because function is not determined by things like degree distribution and clustering and communities, but this is determined by the ability of the system to process signals in a way that those functions are intact and they're doing what they're supposed to be doing. And in a way, I think the control tools offer a layer of tool set that can be applied to an arbitrary network and ask very precise questions about how is that system controlling its behavior and what are the mechanisms responsible for that. And I'm just going to conclude by acknowledging everybody who helped me. This is the team where we did the theory. This is the team of the experiment. And I know why I went that far. That's a different talk. Right? And if there's any time, I'm happy to take questions. I realize that I'm pretty much at my time limit. Thank you.
Gosh, I have one right away. I apologize. I was buttonholed outside, so I didn't hear the first.
But I'm not going to tell you the beginning again.
No, I know. I'll get you later.
Yes,
but no. So it has to do with the end and the 9 neurons that are directing the motors.
Yeah.
Do you know what they're doing?
Well, that's a philosophical question.
No,
right?
no, it's not. There are signals going to the muscles that are giving this very simple behavior, this pattern. Yeah. And there are 2 versions of it. There's forward and backward. Actually 3. Forward, backward, and off.
Yes.
Mm-hmm. Okay. And so that's some kind of rather special signal that these control neurons are sending to the motors.
Correct.
And so what are they?
Good. So first of all, we can construct the signal in the linear approximation, but the C. elegans is deeply nonlinear. So the short answer, we don't know what they're doing. The longer answer is that what we're finding is that the network is wired in such a way that it almost doesn't matter what they're doing. The wiring assures controllability. That's the first step. And the second step is how you build the dynamics over that. And we're not testing at all the dynamical roots here. We're testing the question, is the network wired in a way that controllability at all is possible? So, and that's the primary question. The way to say, like, if you want to get from here to the White House, the first question not is whether you go fast or slow, but is there a road between here and there that is accessible for you? And that's what the network is checking. And the secondary question would be the dynamics, which is a big question in control theory, and lots of people are thinking about how that looks like. We have not answered that. Okay? Yes.
Thanks. This is really fascinating. I'm wondering if you can kind of go backwards. That is, look at an organism that has some deficit in its, some disorder or something in its behavior, and figure out what is wrong in the network, what lack of controllability there is.
I think we can do that in a sense that, yes, the answer is yes, and that kind of leads to another work we have done, which is what we call synthetic lethality. In biology, there is a concept of, like, when you knock out two neurons independently, none of them have an effect, and the two of them together have an effect. So in a way, when I do the first knockout, I'm creating a defect, a disease in the network. And then we do a second one, but it may not be visible to you because it may manifest itself only under extreme conditions that you don't see under normal conditions. And when you knock a second one out that has the same property that it's not visible what happens, but together they have an effect, that's when you actually start getting signals. So we are able to do that. That's a little bit of what you are saying. But the second answer to your question would be, if you bring us a C. elegans that has a mobility problem, and I believe that we can actually, by figuring out what the proper measurements, figure out where the network is broken in that sense.
Just one here. So I have two quick questions. One is, what happens if you break one of these things? Is there a way for one of the neurons? Is there a way for the organism to dynamically repair the network in a minimal number of steps that will lead to controllability? And the second question is, do you know of any systems that are actively trying to avoid controllability so that you?
Good question. So the first one is, it depends on what you mean by breaking it. If you break the network, I believe the answer is no, because the reason why it's no is because the network is built up developmentally in the organism. So if you are actually an adult animal and your network is broken, you typically don't have mechanisms to reconstruct it because the links are forming early in the developmental stage. Now, there are examples of differences and lots of examples of humans, you take half of the brain out and recover certain functions and so on. But I think in the C. elegans, it's such a minimal network, I'm not aware of that to be possible. The C. elegans is genetically pre-wired, meaning that every single animal is the same. If there's a genetic defect that prohibits the formation of one or two links, then there's no way to actually recover that. Now, do we know systems where it's kind of tries to avoid controllability? I don't. I'm assuming that some of the clandestine networks are in that particular space. And aren't we all of us in that space when we try to get away from our bosses? Right? But on the other hand, I don't know on top of my head a really serious experimental system or like real system that we have studied that would have that property.
So you sort of alluded to it at the beginning, but you didn't explain it, and I'm curious. So as I understood it, the Kalman condition mapping onto the max match problem is a function of linear control theory.
Correct.
And what does happen in the nonlinear regime?
Perfect. So this kind of comes back to Bob's question as well in a way that there are two layers to the problem. One of them is that how the network is being wired, and then one is what is the nonlinearity of the system. Nonlinear theory tells us a lot about nonlinear control. And the answer is the following. If you are not messing with the network, but you put a nonlinearity on it, then the predictions from the linear theory will still work as control nodes, except you may not need all of them because the nonlinear modes will kill some of the behavior aspects. So nonlinear systems are easier to control than linear systems because of the decaying modes. So that's number one. The number two is that the type of tools we're using is controllability, meaning that we're checking whether the system is wired to allow for control. And that is independent of the dynamics that we have. If this fails, you cannot make magic with the nonlinearity to resolve that. If this is valid, you can still alter controllability with the net, but the control nodes will remain the same. So in that respect, everything that we do is asking, is the network wired properly for control? And that's independent whether the system is nonlinear or linear.
So you're saying that the nonlinear task is always easier?
Easier in the sense, depending on what you ask, right? So it's easier in the sense that it requires fewer control nodes, but it's much harder because you don't have a closed solution for the signal. So if you come from a control theory perspective and they say, well, but tell me what the signal is, they would say the nonlinear control is very difficult, and where we have solutions for nonlinear controls are specific small systems where things have worked out in a lucky way. And that's where feedback control comes in. So where you cannot really simulate the whole system, you cannot write a closed solution. So I'm just going to look at it, and if you stand up, I kick you down. Right? That's feedback, and you're bypassing that problem.
So, yeah, so just quickly,
Please.
It was very surprising that the degree that the randomizations just with the degree distribution, that was so much of a predictor of the number.
Yes.
And so, could it be that this selection of networks, which I saw it was a very, it covered a lot, but to what degree have you tried topologies? I mean, are there like other aspects of topology that people study, whether it's networks with communities, nest-and-nest, etc.?
Yeah, I hear you.
I was surprised. I mean, do you have networks where the topology is so determined by the degree distribution?
So obviously I presented it as a numerical finding, but this was like 7 years ago. In the meantime, actually, my colleagues at the Rain Institute in Budapest, the mathematicians, have proven to be the case. That is, they have proven that for a large network asymptotically, the number of control nodes will converge to the value that is provided by the matching, and the fluctuations will be minimal around that. That being said, one of the things I learned from my mathematician colleagues, that you can always be counterexamples, right? But those are very pathological cases, and here it's a convergence proof in the sense that if the network gets larger and larger, the number of control nodes converges to the one that is given by the matching.
So you're developing theory for large networks?
That's what we focus on. But in a way, the C. elegans is not a large network, right? And it works just as fine. But the mathematical proof could only work for very large networks.
So I have a quick question. What is the role of stochasticity in all this? Because we know this system is stochastic. So there is something in controllability that have to be related to suppression of noise creeping in the network. Yes,
correct.
So,
so obviously we had this dream that now stochasticity will change everything, and we never published anything because it doesn't. So in the sense that stochasticity is important when you're asking what is the signal that I need to build to control a system, but not whether the network is right for controllability. So none of the effects what we talk about are affected by stochasticity. What will be affected is what we call the control energy, that I haven't had a chance to talk about it, how much effort, how much energy I have to input the system to actually control that system. That will be affected by stochasticity. That will be affected by the weights of the links as well. And we have a formula to tell us what's the energy you need, and that's very dependent on the network structure and so on. But the very question, who are the control nodes, are not affected by that. Perfect, thank you. Thanks.